start also transmitting through YouTube already. Okay, so it's really a great honor for me to present today's speaker. Professor Douglas Higgs qualified in medicine at King's College Hospital in London in 1974 and trained as an hematologist. In 1990, he was awarded a DSc in medicine and is now a fellow of the Royal College of Physicians and Pathologists. He joined the MRC Molecular Hematology Unit in 1977 as a clinical training fellow and honorary NHS consultant. Since 1996, he has been the professor of molecular hematology at University of Oxford. In 2001, he was appointed as director of the MRC MIQ and has focused his research on the processes by which multipotent blood stem cells undergo lineage commitment and hematopoiesis to understand how genes are activated and repressed during normal hematopoiesis and to study the inherited and acquired genetic diseases affecting these processes. The MRC-MI2 is now an international research center of excellence in both red cells and myeloid disorders. Since 2001, he has acted as a chairman of the Department of Hematology and team leader for blood disorders in the Oxford NIHR Biomedical Research Center. This has enabled him to establish an integrated academic center for hematology in Oxford with both a strong scientific and clinical base in red cell disorders, myelodysplasias, leukemias, and lymphomas. Professor Higgs has been awarded with several honors, including the Fellow of the Academy of Medical Sciences in 2001, the Fellow of the Royal Society in 2005, and the Buchanan Medal of the Royal Society for his seminal work on the regulation of the human alpha-globin gene cluster and the role of the ATRX protein in genetic disease in 2013. The main interest of his current laboratory is to understand the fundamental question of how mammalian genes are switched on and off during lineage commitment, differentiation, and development using hematopoiesis as the experimental model. And we'll hear quite a bit about it today. Well, finally, his contributions to human genetic diseases via the hematopoietic system that have been published in top scientific journals, including Science, Cell, Nature, and Nature Genetics, derived from his original and detailed investigation of a single locus, have ideally placed him to direct exciting new studies to understand how the increasingly recognized mutations in regulatory sequences contribute to a wide range of common human genetic diseases. And consequently, he was appointed as the director of the internationally renowned Weatherall Institute of Molecular Medicine in 2012. So, well, with that, I thank Professor Doug Higgs for being here. It's really our pleasure, and I leave the microphone to you.
Lovely. Thank you so much, Mara. That's a very, very nice introduction. And thank you very well to all of you. Thank you very much for the invitation to come. Now, let me just double check that you can see my slides okay?
Yes, we can.
Okay, lovely. So let's start just by looking at what we're interested in the lab. We're interested in how mammalian genes are switched on and off during development, lineage commitment, and differentiation. And we're also interested in how those switches are perturbed in human genetic disease. And then finally, some people in the lab are asking how we can manipulate these switches to improve human health. And so today, I'm just going to really talk about how mammalian genes are regulated. Because I think that's probably more suitable for the audience. Now, as Ira said, we are very interested in looking at globin gene expression throughout hematopoiesis. And of course, you know that starts off with a long-term hemopoietic stem cell that undergoes lineage restriction to form these progenitors. And then the progenitors undergo commitment to form these committed precursors here. And ultimately, you produce the formed elements, the mature specialized cells, which are the formed elements of blood. Now, we're interested in the fact that, or at least thinking about this simply, you can say that essentially this whole process just involves different sets of genes, 20,000 genes, being switched on or off. And again, thinking about it from a simple point of view, I always think, well, we've got the DNA code now, and we could identify the cis elements. And primarily, this is read by the transcription factor program. And then that in turn imposes the epigenetic program, which in turn alters the way that the cell interprets the transcription factor program. But the primary thing reading the genome is the transcription factor program. And we know that because, for example, if we express a gene like GATA1 in this cell, we can make it transfer over to produce this type of cell and go down the erythroid lineage. So it's really the primary readers are the transcription factor program. So our aim then is to understand in detail how genes are switched on and off during this whole process. Now, to try and simplify the problem, we really concentrate on erythropoiesis. So here, this is looking as a stem cell undergoes commitment to form this bipotential cell, which can become either a megakaryocyte or an erythroid cell. And we look at it as it goes down the erythroid pathway and then comes down here to form red blood cells. And just out of interest, I mean, you're forming 1 million of these cells every second, okay? And the good thing about this, pathway is that we can take snapshots of what's going on as the genes are being switched on and off at every point in this pathway. So we can get more or less a kinetic or dynamic picture of what's going on during gene regulation. Now, the genes that we work on are the hemoglobin genes, and you might think, well, they're a rather boring set of genes to work on, but actually they're absolutely perfect for this situation that we're interested in because it's lineage specific. So globin is only expressed down this lineage. And then secondly, there are no downstream targets. And so why is that important? Well, if you could imagine, if you start to work on a transcription factor that's working, say, in these types of cells, then as soon as you alter it, then you can alter the lineage, okay? Whereas with globin, you can alter it as much as you like, and it will always go down this lineage. So that's a really important aspect of looking at gene regulation, because you don't want to look at a gene where the cellular outcome it varies depending on how you manipulate the gene that you're interested in. Okay, so just for those who don't know, I'm sure most of you will know this, but hemoglobin is normally synthesized via two clusters, the alpha-globin cluster and beta-globin cluster, and they come together to form this heterotetramer, hemoglobin A. And our work really concentrates just on the alpha-globin genes. Now, when I was a sort of a young scientist just coming into the lab for the first time, after a year or two, I read this book, which is A Genetic Switch by Mark Potashny. And it had a big influence on me, really, because what Potashny had done is to work out a lot about gene regulation just by studying phage lambda. And this is just a 50 kilobase piece of DNA. And so what I thought was maybe by studying alpha globin expression in its normal chromosomal context, maybe 100 or 200 kb around it, we might understand the principles underlying normal mammalian gene expression, just as Mark had understood how prokaryotes express their genes by studying phage lambda. And so we took that path, and more recently, we've really concentrated on understanding mouse alpha-globin cluster because it provides a really good model of alpha-globin expression, and we can use that in an experimental way, in the way that we couldn't use the human cluster. So this is what we concentrate on. So the first question I really wanted to answer in this sort of part of the work was how much of the chromosome do you actually need to get fully regulated expression? So here are the alpha-globin genes, okay? And you can see that they're surrounded by these widely expressed genes. So how much of the chromosome was necessary to get fully regulated expression? And we did this experiment back in 2007. And if you just took this 80 kilobase segment, we could show that that contained everything that was required to get fully regulated tissue-specific, developmental stage-specific expression of the globin genes. So the next thing we wanted to do was to understand, where the specific regulatory elements were within that. And to do this, we initially looked for DNA's hypersensitive sites. More recently, we used ATAC-seq. And all this does is to show you where nucleosome-free regions are and where transcription factors are binding the chromatin. And what you see is that these 7 regions here are specific to erythroid cells. So we consider they most likely are what's necessary to drive regulation in an erythroid cell. So then we wanted to subdivide these elements. And so we did this in 2 ways. Firstly, we looked at H3K4ME3, which identifies, this is a chromatin modification that identifies promoters in this region. And then we looked at H3K4ME1, and this is a chromatin modification that identifies enhancers. And what you can see is that there's this group of 5 erythroid-specific enhancers that lie upstream of the alpha-globin genes in the cluster. And then we also saw these little peaks here, and those little peaks correspond to Felix's famous protein CTCF. And so you can see that each of those little peaks correspond to CTCF. And so in the end, characterizing this region that we'd identified, it sort of simplified everything because you have 2 promoters, and then you have this group of 5 enhancers, and all of it is surrounded by these CTCF sites. And as you can see, they're orientated largely towards each other. And this is an important feature that you found of CTCF binding elements surrounding a mammalian gene cluster. And so in many ways, this looks like many typical mammalian gene clusters. And so that we thought that if we characterize this, maybe we can understand the principles by which such a cluster might work. Now, the next thing that we've done, and this is the work of Mariika, was to do a self-association map across here. And some of you may know how we do this, some of you may not. All you need to know is that in these heat maps, what we're looking at is which pieces of the chromatin are interacting with each other. And so if you look in embryonic stem cells, when globin genes are completely silent, you find this region is fairly flat, okay? But if you look in an erythroid cell, you see it's a little bit more uneven. So what we find is that in both embryonic stem cells and in erythroid cells, we see these so-called topologically associated domains, okay, here and here. And that just means that within this region, things are interacting with each other a little bit more frequently than they're interacting outside. But as I pointed out, you see a little bit more shape and substance in erythroid cells. And so what's causing that triangle where sequences are more frequently interacting with each other? And it turns out that this is the region that contains the enhancers and the alpha-globin genes. And then this surrounding region here contains the CTCF binding sites. And you can see that in erythroid cells, these CTCF sites surrounding the cluster are now interacting with each other. So this is interesting, and we've been developing that sort of chromosome conformation capture technique a little bit more recently. And James Davis in our lab worked out a way in which we could look very closely at these interactions. So if you capture from the alpha-globin genes here and here, you can see very specific interactions with each of the elements of the enhancer, okay? And even when you go down to further detail, you get base pair resolution of this, and you can see the proteins of the enhancer and in this direction of the promoter, suggesting that when the enhancer and promoter are coming together, they're really in quite close contact with each other, okay? So that suggests to us this type of model in which we have, these are the flanking genes, and in erythroid cells, you extrude this self-interacting domain, and within that, the enhancer and the promoters are coming into close contact. And as I say, we think now, and this is based on the work of Leonid Mirny and Erez Lieberman-Aden, that actually this is happening by an active process in which the loop of chromatin is being extruded. Everything's interacting with everything else. And in particular, the enhancers are in very close proximity to the promoters. So that's a hypothesis, that's a model. And so we wanted to look at that in a little bit more detail. And this is the work of Jill Brown and Veronica Buckle. And what Jill and Veronica did was they looked in erythroid cells and they used fluorescence in situ hybridization probes at the upstream and downstream CTCF binding sites. And then we labeled the region of chromatin in here with a long probe. And so that this was labeled in blue. And what you can see is that in erythroid cells, you got this loop of extruded chromatin where the enhancer and promoter are lying. And then the CTCF sites are actually coming into close proximity. So it's very nicely consistent with the idea of loop extrusion bringing these elements together. It's not proven, but that's the data so far, and it looks quite convincing. So now what's going on inside here? And what the next thing we really wanted to know was, what do the enhancers actually do to influence the transcription cycle? And this is the work of Martin Lark. And so what Martin did was to examine the sort of pre-initiation complex and polymerase II. And what he found was that in the absence of the enhancers, that there's no recruitment of polymerase II to the gene in question. And so what we don't know, because polymerase II is not being recruited, we don't know whether the enhancers might also influence pausing, elongation, and termination. But we know that the first thing it does, most importantly, is to influence recruitment of polymerase II to the enhancers. And I'll show you some more data about that a bit later. So we've carried on, and we want to really understand what goes on within this sort of chromatin domain. And just recently, Ed and Danuta have been looking at how transcription proceeds within that domain. And the way they've done it is to introduce some loops into the alpha-globin gene. And those loops are recognized by a protein which you can label fluorescently. So when the RNA is transcribed, it forms these RNA loops. those RNA loops are recognized by a fluorescently labeled protein, and therefore you can monitor transcription in live cells. And so that's what we've been doing. And what we find is that actually, rather than just being sort of switched on, the gene actually pulses, and it switches on and off with time. So that's really a sort of a rough overview of what we've been doing. And I can then start to sort of put together a model, because we, as I said at the beginning, we've looked at this in each of the stages of erythropoiesis to try and get a dynamic model of what gene expression, how it's switched on. And so the first thing that we find is that lineage-specific transcription factors bind particularly to the enhancers, okay? And then we get loop extrusion, and as part of that, you then get enhancer-promoter interactions. And all of that is taking place within some sort of transcriptional hub. And this is when you get the onset of alpha-globin gene expression. And then as you go a little bit further, that hub becomes consolidated and you get expression. And the way that that happens is by initiation and recruitment of polymerase to the promoter. And the other thing that we've noted just recently is that when it starts off, you get the transcriptional bursting is rather noisy, but then as it goes on and globin expression really gets consolidated, it then becomes rather smooth transcriptional bursting. But it's still bursting at that stage, but it's much less noisy than when we start. And we think that's probably because this transcriptional hub is becoming stabilized, consolidated, and transcription is becoming much more efficient. Okay, so what I hope I've shown you is that the alpha-globin cluster is a really nice model for understanding mammalian gene regulation. It's very similar to many other loci that have been looked at. So I think we can establish general principles from this. And within the lab, we're looking at the role of enhancers, the role of promoters, and the role of these CTCF-bound elements. Obviously, there's not time in one seminar to go through all of that. So I want to tell you about some unpublished data, more recent data, where we've been looking at the enhancers. And again, I think what we're finding is of general importance in mammalian gene regulation. Okay, so the first thing to say is that as with all enhancers, we see these 5 enhancers here. And they are the place where it provides a platform for recruiting the erythroid-specific transcription factors. Okay, so this is probably where the action starts to switch the gene on, and also it accumulates a lot of this protein called mediator 1 or mediator, and this is one component of mediator. And that then is a platform for recruiting all these things. Now, some years ago, Rick Young's lab looked at this type of phenomenon where you get clusters of enhancers, and they ranked these clusters of enhancers using the amount of mediator that was recruited. And as you can see, most enhancers don't recruit so much, but then you get this exponential increase in the amount of mediator recruited. And so they called this group in gray here, they called these super enhancers. And as you can see, the alpha globin and beta globin enhancers are amongst the highest super enhancers that we see specifically within erythroid cells using the criteria set out in this so-called Rose algorithm. So this group of enhancers has subsequently been classified as a super enhancer. Now, the real problem is I still don't know what super enhancers are. And there's a lot of discussion about this. And if you want to read some more about it, you can see that together with Gerd Blobel, we wrote, and Rick Young, we wrote a piece in Nature Reviews Genetics, which really outlines the issues around super enhancers. And then, I think the questions are all super enhancers the same thing? Are they really a new genomic element, or are they just a cluster of regular enhancers? And then this question is rather different. I mean, do they have emergent properties? In other words, is the sum of their activity greater than their individual input? And then the question is, have they been rigorously investigated? And I think, in my mind, the answer to that is no. And so that's what we're going to talk about next. So not so long ago, Debbie Hay did a nice experiment. It took us a long time to do this, but essentially what we did was to delete out each of the enhancer elements one by one, and in combinations, okay? And then from each of those experimental models, we made a mouse and we bred them to homozygosity, and then we analyzed the chromatin structure and gene expression of the alpha-globin cluster. Now, the first thing to say, this is an interesting thing, is that as you take out individual sites, okay, here, 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 and here, and then even when you take out two sites, the other sites, okay, of the elements don't change, okay? So it's not as if these elements are dependent on each other to form their chromatin structure. So that was an important observation. The second observation that we wanted to know was, what was the effect on gene expression? So these really concluded a lot of work, actually, but the first thing to say was that R1 and R2 are the major activators of alpha-globin transcription. So if you take out R1, you decrease alpha-globin expression by about 40%, so maybe 40%. If you take R2 out, you reduce it by about 50%, okay? If you take R3, RM, and R4, even though they look like enhancers, they have very little, if any, activity, all right? So you can take each of those ones out and nothing happens at all. And then the next thing to say is that they're additive, so that if you take out R1 and R2, you get down to 10% of expression. And then if you take out R3 and R2, you get, it's a reduction of 50% and a reduction of 0%, and you still get 50%, okay? So we concluded that these, when you look at the problem this way, that the enhancers are acting in an additive way. Now, when we looked at what underlies each of the enhancers, what we found was that the major enhancers are the most complex enhancers, both in terms of the number of transcription factors, different transcription factors they bind, and the number of sites, okay? So R1 and R2 look quite complex, R3, RM, and R4 look much less complex. And that kind of fits with the expression data. Okay, so then let me just digress for 1 minute, and then to say that actually there's another gene in the cluster called HBAX, and this is an embryonic gene, okay? So what we've been looking at so far is adult erythropoiesis. And what happens if we look now at primitive erythropoiesis? And that gene is now switched on, okay, as judged by this ATAC site and also by gene expression. Now, we made all of the same deletions and looked at them in the embryonic life, and the pattern looks very similar to what you see in adult life. But now, when you look at the effect of these deletions on the embryonic gene, it's quite different. So you take out R1, nothing happens very much. Take out R2, nothing happens. But if you take them both out, you get a 50% reduction in in HBAX gene expression. So the conclusion from that bit is that how these enhancers work depends a little bit on the promoter with which they interact. And that's a new finding, I think. So why might that be? Well, in actual fact, the zeta globin promoter is much more complex than alpha-globin, and it binds many more transcription factors. So maybe it doesn't require so much of the activity of these enhancers as the alpha-globin genes do themselves. Okay, now I don't know about you, but I love reading popular science. And I particularly like this man, Richard Feynman, and I expect you all know that Richard Feynman was the person that worked out a lot of quantum physics and quantum electrodynamics. But he said a very interesting thing, which I sort of took notice of. And he said, "What I cannot create, I do not understand." And so that really prompted us to ask the question, well, could we build a regulatory domain from scratch? Okay, because if we can, then, as Richard Feynman was said, we might understand it a bit better. Okay, so what we did was, here's the normal cluster, and here are the 5 enhancer elements. Here's the embryonic gene and the 2 adult genes. So first of all, we made some ES cells in which we took out the other allele, okay? So that means that everything we look at now comes from this allele. And then we engineered this allele with recombination sites so that we can put any allele we want into that. And together with Jeff Boki, who can synthesize large synthetic loci, we made an allele that has no alpha-globin enhancers in it at all. And then we made those ES cells and then differentiate them into erythroid cells. So what did we find? Let's have a look. So here's the wild type with the 5 enhancers. And here are the alpha-globin genes again. So what happens is that when we take out all the enhancers, as we might have expected, their chromatin accessibility goes missing. They don't bind transcription factors anymore, and there's no modification with H327 acetylation, which is a mark of activation. Now, importantly, what you can see is that the chromatin accessibility at the alpha-globin genes has also gone down considerably. There are no transcription factors binding there. H3K4ME3, which is a marker of, as we've seen, is a marker of promoter activity, is not seen, and H3K27 acetylation is not seen. I'm not showing you all this data, there's too much, but I'm summarizing it here. Okay, so these really look as if they've gone back to what you see in the undifferentiated ES cells. There's just nothing going on here. And so just to let you know that all the CTCF sites in this situation remain fully intact. So what about polymerase 2? What is there any polymerase 2? I mean, could it all be binding there in a paused state, waiting to be released by the enhancers, or what happens? And what you can see here is that in actual fact, polymerase 2 is not recruited in the absence of the enhancers at all. And this goes back to what Martin had found, that the enhancers are absolutely required to recruit polymerase 2. Now, so what we felt is that we've created a blankish canvas, okay? And I say that because in actual fact, there's nowhere in the genome that's a blank canvas. There's always something in there that you can activate or repress, or that will interfere with expression. But we know this region of the cluster very well, of the genome very well, and so we feel that we've got reasonable control over what happens when we either take something out or put something in. So what we wanted to do next was exactly what Richard Feynman had told us to do. We want to rebuild the super enhancer. Okay, so here's what normally happens when you take the elements out. The question is, what happens when you put them back in to this blank canvas? And what we would have predicted, if we put R2 back in, for example, which is the major enhancer, okay, it reduces expression by about 50%. So we would expect that enhancer, because it looked additive when we take them out, we would have expected that to produce about 50% alpha-globin expression on its own. And from our past experience, we know that that should be sufficient to drive alpha-globin expression to make a viable mouse, okay? And so what does it actually look like? So the first thing is it just looks like a typical enhancer when we put it back in. So here you see, here's the R2 only mouse model, and you can see it has a nice ATAC peak, and it binds GATA1, which is one of the main erythroid proteins. It also binds this protein, NFE2. And so you can see that in actual fact, it just really looks like a normal enhancer. And so here you can see histone acetylations there. And here's the R2 only. It's got a little peak of histone acetylation. And then H3K4ME1, the marker of enhancer, you can see there's a little peak of that there. But of course, these active modifications are present at reduced levels, okay, compared with what we would see in the presence of all enhancers. Now, the other thing is that we looked at the important mediator and BRD4 cofactors, and what you can see is that in the R2 only model, okay, you can see that mediator is quite reduced. It's there, but it's quite reduced, as is BRD4, okay, which is another important co-activator. Okay, so the other important thing is that you can see that the R2, just this single enhancer, is able to recruit some polymerase 2, but not as much as we see in the wild type situation. And then we looked at how the R2 element interacts with the alpha-globin gene. So this again is just simply looking at the interaction frequency. And what you can see is if we look from R2, here's what we normally see, okay, in the presence of all the enhancers. And here's what we see in the presence of just R2. So it's there, but it's quite severely reduced. And it's sort of, you know, in between what we see in non-erythroid cells and fully active erythroid cells. And then similarly, if we look from the alpha-globin genes towards the enhancers, we see again a reduced interaction. So the interaction frequency between the enhancer and the promoter is quite significantly reduced. Yeah. Okay, so contrary to our prediction then, we found that R2 homozygotes are mostly non-viable. Okay, so you can pick out the homozygotes quite easily here because they're pale and their livers, as you can see, are a little bit paler than normal. And here you can quite easily pick out the homozygotes that's really quite profoundly anemic. And this is not what we expected. We expected 50% of alpha-globin expression should be absolutely fine. And we only found one survivor. And it had a huge spleen, indicating that it was severely anemic. And what we found was, when we looked at the expression, here we are, this is what we expected to see, and this is what we actually saw. So we're expecting to see 50, 60% alpha-globin expression, and what we saw was only about 10%. Okay, so just to summarize that one experiment, when we put back R2 into that enhancerless landscape, what we found is that R2 creates the mark of an active enhancer. It recruits relatively low levels of co-activators. It does not contact the alpha promoters as frequently. It does not upregulate alpha-globin expression to the degree predicted. And so R2, despite what we thought when we originally just took R2 out of the cluster, is not an independent element, and it suggests to it actually relies on the other elements within the super enhancer. So this is interesting because it's not actually what we predicted at all. So we've gone on to rebuild some other clusters now. So here's what we've done. And we've put a mouse with just R1 in, just R2, and R1 and R2. And all of those mice show the same suboptimal effects, suggesting that R1 and R2, okay, they're enhancers, but they're not as active as they would be if we had the full panoply of enhancers. And so then what we've done is to rebuild all the permutations, and this has been done together with Jeff Bokeh's lab. And so what you can see is exactly the same issue, that in terms of chromatin structure, everything just looks as it would with all the enhancers, but if you take one out, then all the other ones form normally. So it's not acting at that sort of chromatin level. But let's have a look now at what happens to expression. So what we've done, let's just recap on what happens if we take each of the enhancers out one by one or in combination. And this is the work that I've already described. So we take out R1, we get a 40% reduction. R2, we get a 50% reduction. R3, 0, R4, 0. And we think there's about a 10% reduction, maybe less if you take out this last element, R4. And so that adds up to 100%. And then if we take out R1 and R2, you'll remember that we saw a 90% reduction. Now, what happens then if we put them back in? So if we look at their individual expression, if we put R1 in on its own, we get about 12% expression. If we put R2 in on its own, we get about 10%. And if we put R3, R4, or RM in, we get no expression at all. So in actual fact, putting them back individually only gives us about a fifth of what the output is when they're all there. So then we put in R1 and R2, and you can see you get 40%. And then what happens if we put in R1, R2, and RM? It goes up to 75%. And then if we put R1, R2, and R4 in, it goes up to 80%. And then if we put R1, R2, RM, and R4 in, it goes up to 90%. And then if we put everything back in, obviously it goes up to 100%. So we're getting quite different answers from when we take the enhancers out compared with what happens when we put them in. So these two, R1 and R2, look like classical enhancers, but these clearly aren't classical enhancers. And the question is, what are they? They're some sort of facilitating element that is able to allow these two enhancers to be fully expressed. But we don't, I mean, they look like enhancers, but they're not. So what could they be? Well, as probably many of you know, the current feeling in the field is that when enhancers and promoters and so on come together, they form these sort of transcriptional hubs in which everything is interacting with everything else. And there's a very high concentration of transcription factors and cofactors within the transcriptional hub. So one possibility is that these are contributing to forming this type of hub and forming the appropriate transcriptional environment for efficient transcription. That's one possibility. And then the other possibility is that they're really acting as relay elements, so that we've got all the activity here, but to relay it along the chromosome to activate alpha-globin expression, we need these additional elements. I mean, that could be another idea, that they're propagating the signal towards the promoters. So the last experiment I want to tell you about has been a very, very, very long-winded experiment, and Mira has been a wonderful postdoc in the lab, and now she's a sort of senior scientist in our lab. And I must say she's done a wonderful job of doing this. And so what Mira has done is she's taken the normal cluster in which we have this orientation of the superenhancer, and she's asked what happens if we reverse the order of this superenhancer, okay? And what she did was then to make this inversion, she made a mouse model from this. And what you can see is, she calls this the InVen mouse, okay? And so what you can see is that here's the wild type, here's the heterozygote, and here's the homozygote, and this is the spleen. So this is really a sort of a surrogate observation of the anemia that we see. And what you can see also is here's the wild type blood, but if you look at what happens in this case when it's inverted, can you see that you've got this so-called polychromasia, and that's indicative of hemolysis and downregulation of alpha-globin expression. So let's have a look at what happens to alpha-globin expression when Mira inverts this. And you can see that hemoglobin alpha-1 and alpha-2 is quite severely reduced. And hemoglobin X, this is the embryonic, is also quite severely reduced. MPRL3, the gene that contains the enhancers, is unaffected, and these markers of erythropoiesis are largely unaffected. So what it shows you is that if you invert this region, that the expression of genes that normally are activated down here goes down. But if we look at the genes lying upstream, rhomboid and this SNRPN25, what you can see is that both of those genes have been upregulated by inverting the superenhancer. And then here we've looked at the chromatin interactions that occur when we invert the superenhancer. So here's a wild type, and these are the contact interactions that occur throughout the domain. And so you can see that most of the interactions are occurring downstream, and particularly with the alpha-globin promoters. When we invert it, these interactions go down, but these interactions now go up. And here's the differential counts, and you can see that the alpha-globin has gone down considerably, and the genes upstream have gone up considerably. So in summary, I think what we're seeing is that that if you turn this in superenhancer around, then you're changing the direction of its interaction, and this is quite unlike what you see with conventional enhancer elements. Okay, so I think that's really what I wanted to say today, to leave time for discussion. And so the summary is that the alpha-globin locus provides us with, I think, with a lovely model for studying the principles underlying mammalian gene expression. The elements of the superenhancer are not equivalent. R1 and R2 appear to be classical enhancers, but R3, Rm, and R4 aren't classical enhancers, but I think significantly increase the effect of R1 and R2 on expression. And we're trying to work out exactly what role they play. But this observation on the orientation dependence of the superenhancer suggests that they could be acting as sort of relay elements. And we're just checking that observation at the moment. And, just to sort of complete the story is that, we're dissecting the CTCF elements in exactly the same way that we've been dissecting the enhancer elements, and that there are some equally interesting results coming from that. So with that, as I say, hopefully left some time for discussion. I hope I've thanked everybody as we've gone through. But this is all of the group, and I must say it's an absolute pleasure to work with these youngsters who do such a great job and do such nice work, and are lovely people as well. So with that, I'll just say thank you, and let's have a chat about what you think.
Well, thank you very much for this great seminar. I think it was.
I can't hear you again.
Oh, can anyone else hear me?
So sorry, Myra, I can't hear you again.
You can hear you, Myra.
Yes.
Can you hear me?
Yes.
Okay. Oh.
Probably he has his volume down. You may write, if you want, you can write to him that he has to tune up the volume of his speaker, or
Yes.
Hello?
Can you still hear me?
Yes.
You can.
Yes, we
For
can.
some reason or other, it's oh, isn't this frustrating?
Why you don't write the questions and he can answer them?
Yeah, we can do that.
I tell you what, can you give me your telephone number? Just put your telephone number in there, and I'll. Oh no, that wouldn't work, will it? No, because you can hear me, but I can't hear you.
Hello. No. Okay.
I've just. That's it. Can you hear me now?
Hello.
Hello. Yes, I can hear you now.
Oh, great.
Yeah. I'm sorry. There we are. It's worked again.
Oh, perfect.
Okay. Sorry. I haven't heard anything at all yet.
No. That's fine. I'm glad we can hear each other again.
Yeah.
Thank you very much for that fantastic talk. It was really very, very nice to see such detailed work. I really admire that. So I'll just start with a question. It's more that I want to hear your opinion of probably you've seen it, a recent bioarchive by Wendy Bickmore, in which she describes that when enhancers and promoters are less than 100 kb apart, presumably looping is not needed. And I wanted to hear your thoughts on that, because I mean, all this data, at least, well, everything is happening in a smaller scale, which makes me wonder. So I just want to hear your thoughts about it.
Yeah, I mean, this is a very interesting thing, Mara. So there are several reports suggesting that cohesion becomes more important for long-range interactions than for short-range interactions, and several people have highlighted that. I think there are very few observations on it, so I think that perhaps generalizing at the moment might not be quite right. Now, I can tell you one experiment that we've done, which I don't think proves the loop extrusion model, but it makes me think it's quite plausible in the case of alpha-globin. So you've got your 5 enhancers here, and you've got your 2 alpha-globin genes here, okay? Now, what we did was to take a boundary element, okay, and put it in between the 2. Okay, and that boundary element works in an orientation-dependent manner to downregulate alpha-globin expression. Okay, so you put it in facing the enhancers, and alpha-globin expression goes down, and then you put it the other way around, and the alpha-globin expression goes up. So the next thing is that particular the boundary element, which is my nose, if you like, in between my fingers. Actually, where you see that cohesin actually accumulates there, when it's in the direction pointing towards the enhancers. And so what our kind of thought was that what's happening is that, and we see this, that cohesin is being recruited at the enhancers, and then I think maybe traveling towards the promoters. And if you put the boundary in, you see downregulation of alpha globin and upregulation of Rad21 cohesin, banging into that CTCF site. Now that doesn't prove anything. It doesn't prove that it's loop extrusion. And so what we're going to try and do is to do an acute degradation of cohesin to see whether or not that downregulates alpha globin expression. And I think that's the kind of killer experiment. If we can show that's the case, then I think we've probably got good evidence for loop extrusion being important. But and the other thing, Myra, is that it's somehow difficult if there isn't sort of a tracking type of model. It seems very difficult to understand how a boundary element might work, isn't it? Because why would putting something here stop the two things on either side coming together? Who knows what it's going to turn out to be?
Yeah.
But I think one needs to do all of the experiments to work it out properly. And so hopefully, if you invite me back in a year's time, we may know.
We will do for sure. And just another one, I'm taking a bit of advantage here. So in this same sense, have you tried or thought about forcing looping in the context of for instance, the R2, when you have just the R2 or just the R3, and then forcing looping and seeing if that's enough to reestablish expression, for instance?
Yeah, that's a very good idea, and so we haven't decided to do that experiment. So what we've decided to do instead is to bring R2 and R1 much closer to the alpha globin genes. Okay, so if they're acting as relays, then if we bring them closer, then maybe the need for any sort of relay will be abolished. And the question is, if you put them closer, do you get rid of any need for
of course
or something?
I see. Instead of inverting, just bringing them closer and keeping the boundary where it is.
Correct. Exactly so. Yeah. And so those we're expecting the results of that one just before Christmas. So it'll either be a happy Christmas or a very sad one.
Okay. Yeah. Thank you very much. Okay, so we can start with some other questions. Brandon, do you want to go ahead, ask your question?
Yeah, thank you. Hi, Doctor. I think your work is amazing. It was actually very fun to get surprised by almost every slide. But I was wondering, in this model, the globin locus, the globin site, accessibility is dependent. I'm sorry, gene expression is dependent on accessibility, at least in part, right? But that's not true for all the genome. There are sites on which accessibility is not the major influence for the gene for a gene to express, right? So I think I got catched by the idea of super enhancers getting emergent properties, and I was wondering if these elements have the ability to turn gene expression on. But will they be sufficient to turn gene expression on in a site on which accessibility is not the determinant for the gene expression? I'm not sure if that was clear.
Yeah, no, it's just clear. I think so. I'm not sure to what extent. I'm not sure to what extent you're when you see gene activation, you don't see, I mean, how many examples are there where you don't actually see nuclease accessibility associated with gene expression? I'm not aware of genes that do that, but you may be, but I'm not. but I think though, from work both on our work and on the work on the beta globin LCR, that you would find that if you put it into other places, that promoters that were, sort of not normally expressed, I think they will be expressed. Okay, so I think there's a very strong enhancer element to them, and I suspect that they could activate genes that that you wouldn't normally see activated. But the thing is, though, I imagine that what you'll find is that they will be that when they are activated, that then their promoters will become active and their chromatin would become accessible. But we haven't done those experiments yet. But it's a nice thought, and I think they probably are behaving as very strong enhancers and would activate otherwise silent promoters.
Thank you very much.
Thank you.
We have another question. Felix, you want to?
Professor Higgs, as always, impressive, and thank you. It's amazing your work. I have one question and one comment. My first question is, I'm really impressed about your series of experiments in leaf cell imaging, in which you induce the formation of the loops.
Yeah.
Since you have a periodic and like a pulse of formation.
Yes.
My question is, this could reflect part of the extrusion model and or some kind of waves of transcription factors interacting to their binding site and depending on their affinity for those sites?
Yeah. That's
my question. Or maybe a combination of both.
Yeah, so you put your finger on a very good question, and I've just written a grant to try to see if we can understand that, because I don't think we understand at all the dynamics of enhancer promoter interactions and how it relates to transcription. And so what we've done at the moment is to build a model in which we can actually visualize the enhancer and visualize the promoter and visualize transcriptional activation in live cells. So what we're hoping to do is to see the enhancer and the promoter coming together at different times, and to try and work out how those interactions relate to the transcription of the gene using these sort of live imaging approaches. I think your suggestions are entirely plausible, and but I don't think we have the answer to those important questions as yet. But it's a very, very important point.
And my comment is based on some of our recent results in the alpha-globin, in the chicken alpha-globin domain. Yeah. Our data is totally consistent with your proposal that the promoter is dictating the enhancer interaction. Mm-hmm. Because what we have seen in the chicken alpha, the embryonic pi gene, the embryonic alpha chicken pi gene, the promoter is, as you mentioned, is very simple compared to the adult promoters. Mm-hmm. Then in the embryonic stage, we see that what we see is all the domain is in an open chromatin context. Uh-huh. And why is not the enhancer? Our domain is much more simple than, apparently is more simple than yours. But the enhancer, why the enhancer is not activating the adult genes? Because the adult transcription factors are not there.
Yeah, okay.
And then they just activate the embryonic gene. But once the switching is going on, what we have seen is that the complete regions, and including the body genes of the embryonic gene, needs to be DNA methylated as a part of the mechanism. And we have seen that there is a boundary just upstream the adult gene promoters where the DNA methylation ends. Then in this way, the simple promoter is not accessible. The transcription factors for the adult genes are there, and the enhancer, that is the same enhancer for the 3 genes, start to activate the adult genes once the switching is on.
Good. Well, this
And
sounds
this
like
looks for us. This looks for us that the promoter is also very important in those interactions.
I'm sure you're right, and I'd love. Probably I need to sit down and have a look at it with you, and we could have a nice discussion about what's going on. I'd love that. That would be great.
I would like to just comment about this data. Thank you very much.
Well, thank you.
Martí, go ahead.
Thank you. And thank you so much for the talk. I got a couple of questions. You mentioned that enhancers affect differentially a transcription on promoters, and it depends on the promoters. But is this solely this effect solely relies on the sequence of the promoter? In other words, on which transcription factors bind each promoter, or it can also this effect be contributed by things like the distribution of enhancers around the genes, or the distance to the target genes, or other factors? And in line with this question, and just out of curiosity, did you try to scramble the positions of the enhancers? Because I only saw that you put them back into place, but not put them in other places where other enhancers were.
Well, yeah. I mean, you might be right. I mean, we've done a lot of experiments moving enhancers around, and there are always more experiments you could do. I think your question is, do we think that the sequence of the promoter might be the only variable? Well, I don't. It's a question really of whether you keep everything else the same. And if you keep everything else the same, then yes, the sequence probably is important. But then if you start to move enhancers around, it's not the same situation at all. And then, there might be other variables. So, we may have to think about that in the future. But I think we've got enough variables at the moment without making more. but it's an interesting thought. Thank you.
Thank you.
Paula Licona, go ahead, please.
I think Dr. Casillas was before me, no?
Sorry, yeah, that's possible. Lopez Casillas, Fernando, go ahead.
Thank you very much for this excellent talk. I enjoyed it a lot. And now a question more on the general picture and of how these super enhancers may work and might have a functional role in every cell lineage. My two questions are: have you tried to put this whole cassette from the CTF into another hematopoietic cell lineage so that to check whether or not it's really tissue specific? And the other question is, anything like that has been observed for other promoters that are very tissue specific, like the albumin promoter, for example?
Yeah, I mean, so we know it's tissue specific in the sense that if we look at other tissues, none of those ATAC sites or nuclease sensitive sites are found, and none of the transcription factors that bind them and activate them are actually present in those tissues. So this is a highly tissue specific gene. But, I mean, that also applies to many, many other genes that are really only expressed in one cell type. So, but globin's a lovely example of it, but I don't think it's unusual in that sense.
So the same principle could apply for promoters or activation of albumin in other tissues.
Yeah, I mean, albumin's another good example of a tissue-specific promoter. Yeah, yeah.
And have anybody observed any of these super enhancer formation in the albumin promoter?
I don't know specifically about the albumin promoter, but I mean, super enhancers, or clusters of enhancers, whatever you want to call them, are very common in all cell types, and they tend to drive expression of cell-specific promoters, and they drive them at high levels, and they tend to be the genes that are important for lineage specification as well. So, they're extremely interesting at the moment, but we're just trying to understand exactly what they are.
Okay, thank you very much.
Yeah, thank you.
So I'll read a question that it's in the chat by Itzel Alejandra Hernandez. She says, hi, very interesting talk. I would like to know why orientation is so important for the activity of the classical enhancers, why propagation does not occur in both directions.
Yeah, so that's a really good question. So if you look at a classical enhancer, a single enhancer, if you swap it round, it doesn't make any difference to how it interacts. And so, and we've repeated that experiment with our R2 element. So if we put it in either way around, it has exactly the same effect, okay? But if you take the whole thing, the whole cluster of enhancers, and swap that around, okay, all 5 around, then it seems to have some directionality. And that's the puzzle. That's exactly why we're interested in it. So it's not that there's directionality in an individual enhancer, but there does appear to be directionality when you have a cluster of enhancers. And so that's exactly the question. You put your finger on it. That's exactly the question we're trying to answer. Why is that?
Excellent. Paula?
Hi, Professor. Very nice talk. I really enjoy your seminar. And I have a couple of very general questions. The first one is that looking at your data on how busy those enhancers were, like the amount of proteins they have, and your correlation between having more proteins in R1 and 2, my question is whether, in general, in super enhancers, those enhancers within the super enhancers with the highest occupation in protein wise will be the most influential for the transcriptional regulation of the genes, or is just like an observation that's isolated? Do you think that's a general?
No, there's quite a lot of work on this, actually. I mean, it's a good question. There's quite a lot of work on it, and it does appear that the strength of an enhancer is actually related to both the number of binding sites and the diversity of the binding sites within an enhancer. There's a lady who does a lot of this work, whose name just escapes me for the minute, but that, I mean, we made that observation in our original paper in 2016, but it's also been followed up, and now several people have made the same observation, that if you rank enhancers by the number and the diversity of their transcription factor binding sites, you can actually predict to some extent how active they will be as enhancers. So that's, yeah, I think does that answer your question?
Yeah, thank you. And just one more question. So you said, also thinking on your relay model, in which you were saying that R1 kind of can transmit the signal towards the gene. So have you tried in your ESL where you deleted all the super enhancer, just to put the R1 and 2 closer to the gene? That's enough to rescue the transcription, or that doesn't work? So my question actually goes in a sense of, does distance matter in this relay model?
Yeah, no, you're spot on, because that's exactly the experiment we've just done, and that was the one I was hoping to get for Christmas. So you're absolutely right. That's the right experiment to do, I think.
Thank you.
Yeah,
we might write to you to know the answer.
Yeah, I'll send it in a Christmas card. Yeah.
Yeah, exactly. Okay, so there are a couple more questions. I don't know if you have a bit more time.
I have to go in 5 minutes, but
Okay.
I'm happy to answer that, or even if you send them on to me by email, I'd be happy to try and answer them on that way too.
Okay, great. Let's try to have them answered here, and if not, then I can also write your email here, and so that you can answer them directly.
Yeah.
So, Gabriel.
Okay, thanks. I will go quickly in my comment. So I'm not an expert on this field, but I work on protein structure and structure-function relationship. And when you see in a protein non-additive effects like the one that you are describing, what we have learned is that this is the result of non-direct, but on the contacts that are derived from elements that are far away from the sequence, but are close in the three-dimensional space.
Yeah, yeah.
So thinking in your system, I was remembering that the DNA is arranged in nucleosomes, and then these will have to be packed or regulated by other proteins, if I recall correctly. So my question is, the proteins that might be relevant for these non-additive effects that you are observing, are these proteins related to the nucleosome formation, or these are proteins that are relevant for closing together these core elements of the DNA structure?
Yeah, I mean, we'd love to know the answer to those questions, because, I mean, we know some of the proteins that bind each of these elements, and we know some of the interactions that they can engage in. But actually, at the moment, we have a very sort of primitive view of what the proteins are binding and how they interact. What we do know is that if we use chromosome conformation capture type of experiment, that all of those elements are in very close proximity. Okay, but the precise protein interactions that are going on, and you're absolutely right that that's likely to be it must be part of the explanation for why they have the effect that they do. It's early days, I would say.
Thanks.
Yeah, but I mean, we'd love to know that. Yeah.
Okay, great. So, Josue, if you can maybe write your question directly to Professor Higgs, and he can answer like that. Well, we thank you very much for giving this amazing seminar, and we hope to have you around in real life to be able to be together. And if not, we'll surely invite you again to give a virtual seminar. And with that, I think we can end today's seminar. Thank you so much.
Thank you, Mara, and it was a lovely visit. Thank you very much indeed.
Gracias a todos. Buen fin de semana. Bye-bye.
Bye.
Bye.
Bye-bye.